good morning. I am Lakshminarayana Gunta, NEET Zoology faculty at Super 60, Duppulvalsa, Srikakulam. Today, our topic of discussion is classification of animals, particularly the invertebrate phyla. Animals are split into two major groups, the non-chordates and chordates. Chordates, as you know, they possess or they have a notochord in some point of their life, whereas non-chordates, they don't have notochord in their life. Now we are going to discuss the invertebrate phyla. Before we go to this, let us know what is an invertebrate and what is a vertebrate. Vertebrates are those animals having backbone. Okay, invertebrates, they don't have any backbone. Even though some chordates are there, like Urochordata, Cephalochordata, even though they are chordates, they won't possess any vertebral column. Now we are going to discuss the invertebrate phyla. The first phylum is Porifera. Porifera, the word itself indicates the pore-bearing animals. The term Porifera was first coined by Robert Grant, and the animal nature of these poriferans or sponges was first established by Robert Grant. It was also first established by Robert Grant. Robert Grant first coined the term Porifera, and he also established the animal nature of sponges. Porifera includes all primitive multicellular animals. Multicellular means they have been limited to cellular level of organization only. There is no tissue level of organization is found in Porifera. They are the multicellular primitive animals. They are commonly known as sponges. Generally they are marine. Some families like Potamolepidae, Spongilidae, they live in fresh water. Radially symmetrical. Porifera, they exhibit radial symmetry. Radial symmetry means the animals can be cut into two equal halves in a number of planes. In any plane that passes through the principal axis. Some poriferans or some sponges, they are asymmetrical, means they cannot be divided into two equal halves. All sponges, they are sessile. Sessile means they are limited to a particular place and they won't show self-locomotion. And the body wall of these sponges, it is composed of two cellular layers. Outermost layer is called as the pinacoderm. And the inner layer is called as coinoderm. Both these layers, that is the outer pinacoderm and inner coinoderm, they are separated from a gelatinous matrix called as the mesohyl. Here you can clearly observe these are the coinocytes or collar cells which are present surrounding the spongocoel cavity, whereas these are pinacocytes. Here and there are porocytes which allow the water into the spongocoel cavity. Here you can observe a coinocyte, a collar cell. Sponges have water transport system or canal system. The water current system or water transport system or canal system, it is very much essential in obtaining food material, excreting the waste products and obtaining oxygen, etc. And water enters into the spongocoel through these small pores called as ostia. These pores are called as ostia, and these ostia, they lead into the spongocoel cavity. From there it goes out through a big opening called as the osculum. This is the osculum. Okay, and the body of sponges is supported by a skeleton made up of spicules or spongin fibers, or sometimes both spicules and spongin fibers. Spicules may be calcareous or siliceous based on the material. If they are made with calcium carbonate, they are called as calcareous spicules. If they are made with silicon dioxide, SiO2, they are called as siliceous spicules. And sponges are filter feeders, means they feed by straining food particles and suspended matter from the water they feed. That's why they are called as filter feeders. And digestion is intracellular, means the digestion takes place within the cell. In our humans, the digestion is extracellular digestion. Here in sponges, the digestion is intracellular digestion. These sponges, they are hermaphroditic or monoecious, means both the sexes are present in the same individual. Asexual reproduction takes place by means of fragmentation and budding, and sexual reproduction by the formation of gametes. Fertilization is internal and direct development is indirect, involving different types of larvae like parenchyma, trichimella, amphiblastula, which are morphologically dissimilar from that of the adult. Coming to the classification of phylum Porifera, I already told that the spicules, they play an important role in the classification of Porifera. Based on the type of spicules, phylum Porifera is classified into three classes: the Calcarea, Hexactinellida, and Demospongia. These are the three classes, out of which the Calcarea includes all those sponges whose endoskeleton is made up of calcareous spicules. Whereas Hexactinellida and Demospongia, both of them, they include the sponges which are made up of siliceous spicules. Out of this, Hexactinellida include the sponges with siliceous spicules, but six-rayed. These spicules are six-rayed. Already I told you that these are diactin spicule, triactin spicules, 3 rays, 2 rays, and here they are 6 rays. They are called as hexactin spicules and tetractin spicules and polyactin spicules. Here in the Hexactinellida, they bear siliceous spicules and 6-rayed spicules. This type of siliceous spicules are observed in Hexactinellida. Okay, and Demospongia, they also include siliceous spicule, but other than 6-rayed spicules, means here other than 6-rayed, means they may be triactin spicules, tetractin spicules, etc. Calcarea, the sponges, they live in shallow marine waters, and they may lead solitary or colonial mode of life. And the examples include Leucosolinia. This one is Leucosolinia and Sycon, and Grantia. These are the examples for Calcarea. Coming to the next class, second class, Hexactinellida. Hexa means 6. This Hexactinellida includes Euplectilla and Hyalonema. Euplectilla is also called as the Venus flower basket, whereas Hyalonema, it is called as glass rope sponge. They are the examples for Hexactinellida. They live in the deeper part of the sea, and they are solitary, means they lead individually, their life individually. Okay, not in colonies. Coming to the third one, Demospongia. Demospongia includes marine or freshwater sponges. Unlike that of Calcarea and Hexactinellida, which includes marine sponges, this Demospongia includes the freshwater sponges also. The examples for this Demospongia are Spongilla and Euspongia. Spongilla is called as the freshwater sponge, and this Euspongia is called as the bath sponge. Coming to next phylum, that is Coelenterata. Coelenterata is also called as Cnidaria because of the presence of specialized cells, cnidoblasts, called as cnidoblasts. They are aquatic and mostly marine. They are diploblastic organisms. Diploblastic means the cells, they are arranged in ectoderm and endoderm. Mesoderm is absent. First tissue level of organization is observed in coelenterates. First tissue level means in Porifera we have observed the cellular level of organization, whereas here in Coelenterata we can observe tissue level of organization. They are radially symmetric. Here you can observe The radial symmetry means they can be cut into two equal halves in any plane passing through the center axis, and two basic body forms are observed, that is the polyp and medusa. Here you can observe the polyp form and this one is medusa form. And these two forms, they alternate with each other in the life cycle. That's why it is called as metagenesis. And these cnidarians, they bear the specialized cells called as cnidoblast cells or stinging cells or cnidocytes. Okay, these cnidocytes, they are present mostly on the tentacles. And because of the presence of these cnidocytes, they are named cnidaria. These cnidocytes, they are used for anchorage, defense and capture of the prey. Earlier name coelenterata is based on the presence of coelenteron cavity in the center of the hydra or whatever it may be. There is a cavity. That cavity is called as coelenteron cavity. Based on this coelenteron cavity, the earlier name was coelenterata, but now it is called cnidaria. And asexual reproduction is by means of budding. Here you can observe a hydra and it is a small bud developed from the body, and after development it is detached from the hydra and it leads in an individual life. And sexual reproduction is by syngony. Syngony means fusion of gametes. And the development is indirect, involving a free swimming planular larva. Here you can observe the planular larva, free swimming. The cilia, presence of cilia indicates its free swimming mode of life. Okay, phylum Cnidaria is divided into three classes: Hydrozoa, Scyphozoa and Anthozoa. Hydrozoa includes all hydras. Scyphozoa includes jellyfishes. Here you can observe the jellyfish. And Anthozoa includes sea anemones and corals. Okay, coming to the first one, Hydrozoa. Hydrozoa, mostly they are marine. And some hydras, they are maybe freshwatered. And cnidocytes, the stinging cells, cnidocytes or cnidoblasts, they are located on the ectoderm. The examples are Hydra, Physalia, Lobelia. This Physalia is also called as the Portuguese man of war. And this Lobelia is commonly called as sea fur. Coming to the second class of phylum Cnidaria, the Scyphozoa. Scyphozoa includes jellyfishes and all are marine and solitary, means they lead individual mode of life, and all are medusoid forms. Here in Hydrozoa we observed both medusoid forms and polypoid forms. This one is polypoid form and this one is medusoid form. And whereas in Scyphozoa, all are medusoid forms. Cnidocytes, they are located on ectoderm and endoderm, both ectoderm and endoderm. Whereas in Hydrozoa, the cnidocytes are located on ectoderm only. And coming to the examples of Scyphozoa, the Aurelia. This one is Aurelia and this one is Rhizostoma. These are the examples of Scyphozoa. And coming to the third class, the Anthozoa. Anthozoa includes anemones and corals. Anthozoa, all are marine and only polyp forms are observed. In Scyphozoa, all are medusoid forms. In Anthozoa, all are polypoid forms. In Hydrozoa, both polyp and medusoid forms are there. And the cnidocytes located on ectoderm and endoderm. Unlike that of Hydrozoa, cnidocytes are present in ectoderm and endoderm. The examples for this Anthozoa includes Adamsia, Metridium, Pennatula, Gorgonia. This Pennatula is also called as the sea pen, and this Gorgonia is called as the sea fan. Coming back to the invertebrates, the next phylum is Ctenophora. Ctenophora is also called as Cnidaria, as they don't have cnidocytes or cnidoblasts. These are some illustrations of ctenophores, and these ctenophora, they are known as sea walnuts or comb jellies or sea gooseberries. Sea walnuts or comb jellies. These are considered to be the close relatives of cnidarians, as they are also limited to tissue level of organization, and most of the characters, they are similar to cnidarians. They are considered as the close relatives of cnidarians. These ctenophores, they are exclusively marine. Like that of echinodermates, these ctenophores also, they are exclusively marine. They are diploblastic. They are diploblastic, means there are two layers, ectoderm and endoderm is there, and mesoderm is absent. Tissue level of organization is seen as like that of cnidarians. And the particular character of these the ctenophores is the body bears 8 external rows of ciliated comb plates. Ciliated comb plates. Cteno means comb. Comb plates are there. That's why they are called as ctenophores. These 8 comb plates you can observe here in the diagram. Okay, these are comb plates, 8 in number. These comb plates are 8 in number. And the most important thing with these ctenophores is they exhibit bioluminescence. Bioluminescence is well marked in ctenophores. Bioluminescence means is a phenomenon emitting of light by a living organism. Okay, and there are specialized cells. The cells are lasso cells or coloblast cells. These are glue cells that are helpful in capturing the food. The sexes are not separate. That's why they are called as monoecious or hermaphrodite. Reproduction is only by sexual means. Reproduction is always by sexual means. Fertilization is external with indirect development, includes a larva called as cedipede larva. Most important larva, cedipede larva. Okay. Based on the presence or absence of tentacles. Ctenophores are classified into two classes. Tentaculata, nuda. Tentaculata, it includes the adults with 2 aboral tentacles. Whereas nuda won't bear any tentacles. Even the larval stages also, they won't bear any tentacles. Coming to the first one, tentaculata, means they bear 2 aboral tentacles. These are the representatives of tentaculata: Pleurobrachia, Hormifera. This here you can observe a pair of tentacles in Pleurobrachia. Because of the presence of a pair of tentacles, this one they exhibit biradial symmetry, not radial symmetry. Biradial means it can be cut into 2 equal halves only in 2 planes that passes through the principal axis. And this is Hormifera. And coming to the next class, Nuda. It includes the ctenophores without tentacles. The Nuda, the name itself indicates. Examples are Bero. Here you can observe 8 comb plates, but there are no tentacles. Means all those ctenophores without tentacles are included in the class Nuda. Okay, thank you very much. And in our next class we are going to discuss the Platyhelminthes, Aschelminthes and Annelida. Thank you.